This is the pre-recorded lecture for Thursday, October 1st, 2020 for BIOL 113. This does not include the slides on flagellar motility. That was in a separate mini lecture recording that I'd like you to watch in conjunction with the lab part of the class for week 2. But we're going to talk a little bit more about other mechanisms of motility here, as well as talking about the other common structures on the surface of bacterial cells. Okay, so there are strategies for motility without flagella. These typically involve, though, or are limited to moving on a surface, which is a common context for bacteria to be living and growing on. So it's not that unusual. In fact, it's probably more common than living in an aquatic environment. Now, we did talk about swarming motility on an agar surface using flagella, but these examples could be carried out on an agar surface without using flagella. So some bacteria use so-called gliding motility. This involves sort of sliding over the surface in a way that's not very well understood. But one of those processes, at least, has recently been shown to use structures called pili. Now we're going to talk about pili a little more in just a second. So think of this as sort of a preview of that. These are bacteria. The video I'm going to show you right now is in a group of bacteria called the myxobacteria, which live on surfaces in soil typically. So this is going to be using a type of pilus to basically pull the cells along a surface. So I'm going to switch here.
As seen in this movie.
Just a second. I'm going to switch here to a different screen to share. Do full screen on this. Now, this is actually from one of the papers that some of you are going to be talking about later on in class today. Okay, so hopefully you've seen this video if you're presenting that paper, because I think this video really sort of captures the essence of what they did. And I'm not going to show it all to you right now because I'm going to let the people doing that paper talk about most of it, but I just want to show you the basics of it.
With a light microscope, Myxococcus xanthus cells glide forward in a mode that has been called.
So this is in a phase contrast microscope, and these cells are actually on a very thin layer of agar, and you see they're sliding on the agar. These cells don't have any flagella. You couldn't tell here because you couldn't see the flagella in the phase microscope anyway, but there's lots of other evidence that they don't have flagella. So they're sliding, often in groups, on the surfaces of agar.
Social motility is driven by a molecular machine called the type 4 pilus, which projects forward, attaches to a cell or surface, and then retracts.
So these are not pili. I'm sorry, these are not flagella, these are pili. And so they're extending out from the pole of the cell, attaching and anchoring, and then retracting to pull the cell along.
Type 4 pili can be visualized directly in 3D in a near-native state through electron cryotomography.
So this is a fancy version of cryo-electron microscopy that they call cryo-electron tomography. And this is looking at the base of these pili and then averaging a bunch of images together to get a structure.
Basal bodies without pili can also be seen. Aligning and averaging many of such 3D images results in a clear picture of the machine. Using diverse information about components, including atomic models where available, a pseudo-atomic model of the assembly can be proposed. Our current working hypothesis is that in the cytoplasm, the AAA ATPase pill.
All right, I'm going to stop it there. If Well, if you're working on this paper, you'll be explaining this to other people. If you're not working on it, then you'll be hearing about it from the students who are working on it. Okay, so, but you have the link to this video too, so you can go ahead and watch the rest of this too. I find this to be incredibly interesting and impressive work. And as you're thinking about it, compare it to how the flagellum grows, because there are some striking differences. There are some parallels, but there are also some striking differences between the pilus, how the pilus grows, and how the flagellum grows. Okay, so I'm going to go back now to the PowerPoint. So that's one example. The cyanobacteria that carry out gliding motility do not do it the same way. They don't have these kinds of type 4 pili. And so this is another mystery that has yet to be solved, how these cyanobacteria glide over the surface. There are other bacteria that carry out a process called twitching motility that also uses pili, but apparently not in the same way that myxobacteria do. So similar, but not identical to gliding motility. So those are two examples of how bacteria can move without actually having flagella. Here's another really interesting example that comes from a couple of pathogenic bacteria. And this is movement inside a host cell. These are both bacteria that happen to cause severe diarrhea. So the bacteria are acquired in food or water, they get down into your intestines, and then they actually invade these intestinal epithelial cells lining the wall of your intestine, and then basically hijack the host cell cytoskeleton to propel themselves both within the cell and between cells, as you see here, penetrating from the initially invaded cell after they've replicated into adjacent cells. So sort of spreading along the lining of the intestine in a place that is protected from your immune system, right? So not only are they not getting basically pushed out through the diarrhea, but they are also not accessible to antibodies and phagocytic cells that are trying to attack them. So this is it's a great niche for them, lots of food, very unfortunate for us. Now, I'm going to show you a video of this in just a second, but the thing I want to point out to you is that Shigella dysenteriae is basically an E. coli. All Shigella are just modified E. coli, essentially. So it's a gram-negative bacteria. Listeria monocytogenes is a gram-positive bacteria. So Listeria is famous for food poisoning. Shigella more often it's waterborne, but it also is transferred in food. There was an outbreak of Shigella in San Jose a couple of years ago from salsa, I believe, in a Mexican restaurant. But at any rate, Listeria, so the point is these two bacteria are very distantly related, but they independently evolved the same mechanism of moving inside cells, which is to hijack the actin microfilaments in the cytoskeleton of these cells. So let me click on this and show you this for just a sec.
This mammalian cell has been infected with pathogenic Listeria monocytogenes. These bacteria move throughout the cytosol by recruiting.
All right, hold on. Here we go. All right, now we have the cell. So this is a cultured human cell here, and the bacteria are these dark ones. So this is actually a great slide for showing the relative sizes of the bacteria. But the other thing I want you to recognize here is the sort of shadow at the ends of these Listeria cells. These are actin, these are multiple actin filaments here. So the bacteria is nucleating the formation of these filaments, and then essentially as the actin subunits come on, it's pushing the bacteria across the cell.
including
Okay.
host cell actin, which polymerizes and pushes them forward, producing a comet's tail in their wake. Whenever a bacterium is pushed into the plasma membrane, it creates a temporary protrusion and is then bounced back to continue its random path. If we look closely, we can see a bacterium divide inside the host cell.
Okay, you can watch the rest of this on your own time. Again, this is a very cool
Immediately
video.
after separation,
Sorry, stop sharing here. I'm going to share again and go back to our presentation, right? So the idea here is that this motility allows the cells to go from, allows the bacteria to go from one host cell to the next, okay? So actin-based movement, taking advantage of the host cytoskeleton. All right, other structures on the surface. Now we already talked about pili for their role in motility, surface-based motility in some bacteria. That is generally not their main function, broad function across the bacterial world. What they are designed to do is basically to stick onto surfaces, right, to be used for adhesion. And that's often important in pathogenesis when you want to find your target niche in the host so that you can bind to it. The tips of these pili often have specialized adhesins that will bind to, for example, glycoproteins or on the surface of host cells and recognize them fairly specifically. Now, it doesn't have to be on a host cell. It could be in the environment too, on inanimate objects, other things, right? A special version of the pilus is the conjugal or sex pilus that is used for basically connecting one bacterial cell to another for the passage of DNA between them. So there are plasmids called conjugal plasmids that encode the proteins to make up this pilus. Basically, it reaches out there when it contacts another one. It will actually contract to bring these cells closer together, and then there will be a hollow channel through the middle that the DNA will pass through when these cells are connected to each other, so that a plasmid can spread itself laterally in the population from one bacteria to another. They may be the same species, they may be a related species. It doesn't have to be just moving from one cell of the same species to another. Okay, so those are conjugal pili or sex pili. Okay, so moving on. There are other ways that bacteria can adhere to a substrate as well. Remember I was telling you about Colobacter when we were talking about the structure of the nucleoid, and let's see, gosh, that was only, it's today is Wednesday. That was recently. We'll just say that was just last Thursday. Okay, so anyway, Colobacter has a stalk at one pole. So this is sort of a specialized extension of the cell envelope at the pole. The best one to look at is right here. And at its tip, it also has an adhesion called a holdfast that these Colobacter use to anchor in the environment. These are not pathogens. These live in freshwater marine environments. They attach to the substrate or to shells, whatever. When they're not flagellated motile cells, they are attached stalked cells. And in fact, the length of this stalk can be controlled by nutrient concentration, so that if the bacteria is starving for certain nutrients like phosphorus, for example, it will actually extend this to give more room for nutrient uptake, because this is a legitimate cell envelope, just much narrower. So there's no like nucleoid inside the stalk there, but it can still have transport systems in it. So it can use that to basically increase the surface area of the cell with a much less dramatic increase in the volume of the cell. Okay, So that is the adhesive stalk in some bacteria. Now, again, thinking about ways that you can attach to surfaces, because there are lots of reasons bacteria may want to do that. This is a way that doesn't involve an extension of proteins or the cell envelope. These are polysaccharide capsules, right? Now, if you've done lab already this week, you looked at something like this, which is a capsule stain. Essentially, it's a negative stain. So this is where all the stain is, and it is not in the capsule. So you can see this clear area surrounding these strep pneumoniae cells, right? And so these actually have a sugar coating on the outside, kind of like an M&M, except it's a lot thicker, right? So this capsule, in pathogenic bacteria like streptococci, its main point is to protect against the host immune system, because antibodies, for example, can't get in there, and the complement proteins have a hard time getting in there to access the surface of the cell. And we're also not as good at making antibodies against sugars and polysaccharides. So it's not as antigenic as other molecules on the surface proteins would be, okay? But it can also just serve as an adhesant. Of course, it can do these things at the same time. And bacteria that make these capsules are often very good at forming biofilms, so surface attached communities that have this sort of extracellular matrix surrounding them. And just to throw this out there, capsule formation, so strep pneumoniae is an important pathogen. It's a major cause of bacterial pneumonia, not the only cause, but one of the most important ones. And about 80 years ago now, scientists at the Rockefeller Institute in New York City were studying this in a set of experiments called the pneumococcal transformation experiments. I'm not going to go over this now. We'll see this more in chapter 7, but remember that, because this was a very famous experiment. It was the first evidence that DNA could serve as the genetic material, and it used these pathogenic streptococci that make capsules to protect themselves from the immune system. All right, so this is just reiterating what I was just telling you. Biofilms are very important in natural environments, whether inside a host or outside of a host. A great place in our own bodies to see biofilms is dental plaque, right? The stuff that gets on your teeth, especially if you don't brush your teeth for a while. We get this sort of layer over the top of our teeth, which is basically bacteria growing there and aggregating particles from the food we eat and secreting extracellular polysaccharide. And if we don't clear off this dental plaque, eventually these bacteria, which are, a lot of them are other kinds of streptococci, particularly Streptococcus mutans, will, through the process of fermenting the sugars in our diet, they will produce organic acids that will break down the enamel of our teeth and cause cavities. So there are lots of contexts where biofilms are very important. Dental plaque is just one of them, but I know several of you are interested in dental school. So this is a fascinating topic that a lot of people are studying. Okay, I'm going to wrap up with one last type of surface structure on the surface of cells. These are called S layers or surface arrays. Not all bacteria have this, but some do, and this is relatively understudied. Essentially, in this scanning electron micrograph, you can see on the surface of this particular cell, I need to look up what kind of cell this is. I don't remember off the top of my head. This sort of regular meshwork, it literally looks like chainmail on the surface of this bacteria. This is a protein-based mesh, this S layer. And the best we can tell, its most common function is to protect the bacteria from viruses. Viruses that infect bacteria are called bacteriophage. So to protect bacteria from bacteriophage, keep them from getting access to the surface of the cell, or to protect them against some other types of predators that may eat bacteria. This includes other parasitic bacteria like the Delevibrios. So surface rays are a really interesting topic that has not been studied too much. So I'm going to end it there. Again, in class, we're going to do presentations of research articles in small groups, but I'm going to stop there with this lecture. Okay, going to now